Hi, my name is Pamela Coons, Associate Professor of Medicine in the Division of Oncology at Yale School of Medicine and Yale Cancer Center. I'm excited to announce ASCO's new open access journal, JCO Oncology Advances. As the inaugural editor-in-chief, I hope to support JCO Oncology Advances to become the premier platform to bridge the gap between accessible scientific research and clinical care. Stay tuned for more information, including new article types, at ascopubs.org/JCO Oncology Advances. We look forward to seeing your submissions in spring of 2024.
This JCO podcast provides observations and commentary on the JCO article, Single-Agent Lenalidomide in Patients with Mantle Cell Lymphoma Who Relapsed or Progressed After or Were Refractory to Bortezomib, Phase 2 MCL-001 eMERGE study by Andre Gua and colleagues. My name is Brian Till, and I am an assistant professor at the University of Washington and an assistant member at the Fred Hutchinson Cancer Research Center in Seattle, Washington. My oncologic specialty is lymphoma. In the article that accompanies this podcast, Dr. Gua and colleagues report the results of a large multi-center Phase 2 study testing the efficacy of single-agent lenalidomide to treat patients with relapsed or refractory mantle cell lymphoma. Lenalidomide is a synthetic derivative of thalidomide, and although the mechanism of action is not well defined in mantle cell lymphoma, it has been demonstrated to have multiple immunomodulatory effects such as augmentation of T lymphocyte and natural killer cell activity, inhibition of regulatory T cells, and upregulation of co-stimulatory ligands on tumor cells, and the drug also has anti-angiogenic and direct anti-proliferative activity. Lenalidomide is therefore expected to have little cross-resistance with other active agents used to treat mantle cell lymphoma, and is therefore potentially an important weapon in the arsenal for this disease, in which relapse remains a major problem and for which new therapies are clearly needed. Patients in this study were required to have been previously treated with rituximab, cyclophosphamide, and either an anthracycline or mitoxantrone, drugs that are included in the most historically common induction regimens such as R-CHOP and R-hyper-CVAD, and patients also had to have relapsed or refractory disease following bortezomib. The patients then received lenalidomide 25 milligrams orally on days 1 to 21 of each 28-day cycle until progressive disease, intolerance, or voluntary withdrawal. Primary endpoints were overall response rate and duration of response. Prior to this large Phase 2 study, 3 smaller studies investigated the use of single-agent lenalidomide in relapsed or refractory mantle cell lymphoma and showed response rates of approximately 30 to 50%, response durations of 13 to 22 months, and progression-free survival of 4 to 6 months. In the current study, the overall response rate in the intent-to-treat population was 28%. The median duration of response was 16.5 months, and progression-free survival was 4 months, results which were comparable with the previous studies. Although these outcomes are somewhat modest, these patients were heavily pretreated, and the minority of patients who did responded quickly, with a median time to response of just over 2 months, and responders had prolonged remissions, more than 2 years in some cases. Treatment was generally well tolerated, although 19% of patients discontinued treatment due to adverse events, and only 60% of patients completed 3 or more cycles. Most grade 3 to 4 toxicities were hematologic, but there was a 7% rate of pneumonia and 7% rate of thrombotic events. There was no obvious increase in secondary malignancies, but the median follow-up was short. And since multiple myeloma patients treated with lenalidomide have an increased risk of secondary malignancies, longer observation will be needed to assess this. So what does this study add to our knowledge of the treatment of mantle cell lymphoma? Although it is not the first study to assess lenalidomide as a single agent for relapsed mantle cell lymphoma, it is by far the largest and thus provides the most accurate estimates of activity, at least in the population of bortezomib-treated patients. Although it was a single-arm study, it was a well-designed and well-run prospective study with central review for response assessment, multi-center patient accrual, and thorough reporting of study methods, outcomes, and toxicity results. For any given disease, the practicing physician would ideally like to have evidence-based guidance for making clinical decisions for every situation he or she encounters in the clinic. In diseases where heterogeneity exists in the treatment, prognosis, biology, or other variables, to achieve this goal, there would need to be a multitude of studies addressing each clinical scenario, and these studies would have very focused and narrow inclusion criteria in order to answer each question in a scientifically rigorous and precise way. However, since patients and resources are limited, such a comprehensive spectrum of trials is not possible, particularly for rare diseases. So, in designing trials for these diseases, there's an inherent tension between scientific precision and real-world applicability. Should investigators limit the study to a narrow subset of the population, making the study more scientifically precise, or should the study have broader inclusion criteria in an effort to make the results more applicable to all patients with that disease? Either approach has its drawbacks. In the current study, the authors chose a more narrow approach to eligibility and should be congratulated for successfully completing a relatively large study in a rare disease that did answer a specific therapeutic question for a defined subset of the population. However, given the reality that other studies are not likely to be performed to assess the response of lenalidomide for other specific subsets of relapsed mantle cell lymphoma patients, such as those who receive bendamustine induction rather than an anthracycline-based regimen, or who have not had prior bortezomib, or who have had a prior allogeneic stem cell transplant, this narrow focus, while scientifically commendable, may come at the expense of some real-world applicability, and thus broader inclusion criteria may have made the study more relevant to the practicing clinician. Another practical consideration is that this large study provided the basis for FDA approval of lenalidomide in mantle cell lymphoma, but due to the narrowly defined eligibility criteria, the indication is limited to the subset of patients who relapse after bortezomib rather than the much larger pool of all relapsed mantle cell patients who could potentially benefit from the drug. In conclusion, despite the limitations noted, this study confirms the activity of single-agent lenalidomide in relapsed mantle cell lymphoma and leads us to consider the possibility that perhaps lenalidomide in combination with other agents, particularly antibodies, antibody-drug conjugates, or small molecule inhibitors, could lead to more effective therapies for a patient population that clearly needs them. Ongoing clinical trials are evaluating lenalidomide combination therapies, such as lenalidomide in combination with rituximab as maintenance therapy after induction, or lenalidomide with rituximab and/or bendamustine in the frontline setting, and we eagerly await the results of these studies. This concludes this JCO podcast. Thank you for listening.
For more original research, editorials, and review articles, please visit us online at jco.org. This production is copyrighted to the American Society of Clinical Oncology. Thank you for listening.